Organs of the digestive system are divided into two categories. Alimentary canal, all parts of the same long tube, and accessory organs attached to the tube. These organs participate in the functions you see listed here: intake, putting food stuff into your mouth and keeping it there; breakdown, smashing the food stuff into smaller pieces, or breaking large molecules into small ones; absorption, moving the small molecules from the tubular organ into the blood and lymph for use; elimination, taking the leftover waste and removing it from the body. Along the way, each organ has to time its motility and secretions. So this means multiple levels of regulation, which is organized into 3 phases. The organs of the alimentary canal run from the mouth to the anus: oral cavity, esophagus, stomach; small intestine, large intestine. They are associated with intake, breakdown, absorption, and elimination. Accessory organs include tongue, lips, cheeks, salivary glands, teeth, gallbladder, liver, pancreas. They are associated with breakdown and secretions. An overview of the digestive system. Let's look at each function and the organs associated with that function in a little more detail. Ingestion. The oral cavity and its accessory organs allow you to put those foodstuffs into your mouth and to keep them there. Mechanical digestion. Breaking larger particles into smaller particles. This is not chemistry, but rather smashing and grinding. Mechanical digestion takes place in the oral cavity with chewing, in the stomach during its specialized movement called churning, and even in the small intestine with its specialized movement called segmentation. Propulsion. Moving the foodstuff. So this happens throughout the canal. Note the organs on the diagram. Propulsion moves the foodstuff to the next organ in the canal and includes peristalsis, or wave-like contractions. Note, there is such a thing as reverse peristalsis. That's what happens when you vomit. Chemical digestion. This is chemistry. Breaking larger molecules into smaller ones. Chemical digestion of starch begins or starts in the mouth, but chemical digestion of proteins and fats begins in the stomach. This molecular event is completed in the duodenum and jejunum, the first portions of the small intestine. Absorption. Moving the newly formed small molecules across a vessel wall into body fluids like lymph or blood for transport. Defecation. That's pretty much elimination. There are 2 forms of large movement. Peristalsis, again, moving the foodstuff with wave-like contractions to the next organ. Segmentation, a functional sectioning of the organ, mostly in the small intestine, that allows for mixing contents. Once mixing is finished, the small intestine will then switch to peristalsis to move the foodstuff forward. So all this moving, secretion, digestion takes regulation. The big picture here is the digestive system primes the next organ in the chain. For example, think about food and your mouth gets ready. Chew food and your stomach is getting ready. Have food in your stomach and your intestines are getting ready. To do this, requires homeostatic control by the endocrine and nervous systems. Here we see some of that control. There are two forms of stimuli, mechanical and chemical. Mechanical stimuli is usually an organ stretching because foodstuff has entered it. Chemical is exactly what it sounds like. The chemical components, substrate molecules, osmolarity of the foodstuff are detected and stimulate a pathway of some sort. With regard to nervous system control, we can use extrinsic systems like the parasympathetic nervous system and the vagus nerve. But we will also use a localized nervous system within the alimentary canal. This intrinsic system is called the enteric nervous system, sometimes called the gut brain. A diagram of the extrinsic and intrinsic control methods. Take some time with this diagram and note the different types of receptors. Also note reflex types. As we travel through the alimentary canal, we will discuss lots of reflexes. If the reflex involves the parasympathetic system, that's a long reflex. But if it is localized within the organ using the enteric system, that's a short reflex. A little more about the gut brain or enteric system. There are two subdivisions. The submucosal division controls muscles of the mucosa, the inside lining, and mucus glands. The myenteric division controls the big muscles doing segmentation and peristalsis. Also, this can carry long reflexes. All organs of the alimentary canal have 4 layers or tunics in their walls. You can see black boxes around these layers and their sub-layers. Mucosa. This layer touches the lumen and contains 3 sub-layers of different tissue types and function. Submucosa. Deep to the mucosa. It is primarily connective tissue and houses glands, large blood vessels, and nerves. Muscularis externa. 2 or 3 sublayers of smooth muscle sheets. These sheets of muscle tissue lie on different planes and so contract in different directions. It is the coordination of these sublayers that causes peristalsis, segmentation, or churning. Serosa or adventitia, the outermost layer that is thin. It will house major blood vessels or nerves. When the 2 sublayers are present, it is called a serosa. However, the esophagus is the exception of the alimentary canal organs. Your esophagus lies in a completely different body cavity and does not have the visceral peritoneum wrapped around it. Because of that, the structure of this layer is a little different, so it gets a different name. In the esophagus, the outermost layer is called the adventitia. A-D-V-E-N-T-I-T-I-A. The 3 sub-layers of the mucosa. Epithelium. In contact with the lumen, its tissue is simple columnar and contains goblet cells. This is where secretion and absorption will take place. Note, except in the esophagus and rectum. There, the epithelium is stratified squamous. A mucus is supplied by large glands. Lamina propria. Areolar and reticular connective tissue proper. This sublayer contains the blood vessels for the epithelium. Muscularis mucosa, smooth muscle responsible for localized movements of the mucosa only. The submucosa, dense and elastic connective tissue proper. This layer contains the larger vessels, lymphatic vessels, Peyer's patches, and larger nerves. The enteric plexus or gut brain is also located here. Muscularis externa, again, 2 or 3 sublayers depending on the organ. The innermost layer is oriented around the lumen and is the circular muscularis externa. If this contracts, that spot on the organ will become smaller in diameter. Think of this as a squish, like squeezing on a tube. The outermost sublayer is the longitudinal muscularis externa. Its orientation is along the length of the organ, so when it contracts, it shortens the tube. Think of this as a push. Peristalsis, an alternating push and squish along the tube in a wave-like fashion. Segmentation, a section of the tube pushes and squishes back and forth until mixing is completed. Now, the third sublayer is only in the stomach. It allows the stomach to churn and is called the oblique muscularis externa. Serosa, again, the outermost layer that contains the visceral peritoneum for the abdominopelvic cavity organs. Note, when an organ lies outside the peritoneum, it is referred to as being retroperitoneal. We can use this term to describe the esophagus and anus. The 3 phases of digestion: cephalic, gastric, intestinal. Again, all this moving, secreting, absorbing, mixing requires a tremendous amount of coordination by endocrine and nervous systems. We can stimulate digestive processes as well as inhibit them. Cephalic phase, stimulated by food. Think about it, smell it, touch it, or taste it. Your cerebral cortex receives the stimuli and does a couple of things. 1. Fires long reflexes via cranial nerve pair 10, the vagus nerve, to your stomach to stimulate gastric secretion and motility. And 2. Stimulates salivation in the mouth. The gastric phase is stimulated by a bolus, or a ball of chewed up food, that enters the stomach. This stretches the walls of the stomach and fires the reflex to start this phase. The gastric phase increases the stomach's activity. It starts to secrete more and move more. Its enzymes, acids, and movement will liquefy the bolus into something we now call chyme. Once chyme is formed, a little will leak into the small intestine, and this will trigger a change in events. The stomach will stop its churning and secreting, open a sphincter, and switch to peristalsis. Intestinal phase, stimulated by chyme entering the duodenum. That's the small intestine, the first portion. The chemical makeup of the chyme is detected, and short and long reflexes fire. The intestinal phase stimulates the small and large intestines and changes the stomach's behavior from churning to peristalsis. Now, let's focus on the functionality of each alimentary canal and accessory organ in turn. The oral cavity is responsible for mechanical and chemical digestion of the food you eat. Once that's completed, you swallow the bolus using your tongue and pharynx. To begin chemical digestion of starch and lubricate the bolus, you'll need saliva produced by the salivary reflex. To swallow the finished bolus, pushing it past the pharynx into the esophagus, you'll need the swallowing reflex. Our tongue. It helps with mechanical digestion and swallowing. The tongue does have taste buds that are microscopic and seen in lab. However, it also has small bumps or papillae. For fungiform and circumvallate do have the taste buds on their sides. The filiform don't have taste buds and are more for gripping. The next time you chew, pay attention to what your tongue is doing. Salivary glands. Larger extrinsic glands are the parotid, submandibular, and sublingual. Do note their locations, as you'll also see them in lab. There are small ones scattered throughout the oral cavity called intrinsic salivary glands. Now, obviously, these glands secrete saliva. Saliva cleans the mouth, moistens the bolus, and contains enzymes that will start chemical digestion of starch. A little more about saliva. It is mostly water, so hypoosmotic. It contains a lot of stuff. You don't need to memorize everything here, just the categories. Saliva contains a lot of electrolytes, some digestive enzymes, especially salivary amylase, which breaks down starch, which is called amylose. It also has some proteins. Mucin makes it thick and slippery. Lysozymes, immunoglobulin A, and defensins are for immunity. There's also waste molecules like urea in it. Saliva production is reflex controlled. See, smell, taste, think about food, and your mouth waters. That sensory information is perceived by the cerebral cortex. It fires a pathway through the hypothalamus to the cranial nerves that control the extrinsic salivary glands. The salivary reflex travels on these two cranial nerves, cranial nerve pair 7, facial, and cranial nerve pair 9, glossopharyngeal. Note the specific glands controlled by each cranial nerve. Once a bolus is formed, it needs to be moved to the esophagus. To do this utilizes the swallowing reflex, which is really two reflexes. The first part, buccal reflex, is voluntary or somatic using skeletal muscles. You decide to swallow. The tongue pushes the bolus back to the oropharynx. Your soft palate raises to close off your nasopharynx, and skeletal muscle contractions push the bolus down into the esophagus. The buccal reflex automatically triggers the pharyngeal esophageal reflex. This is involuntary or autonomic, using smooth muscles of the muscularis externa to trigger peristalsis throughout the rest of the esophagus. A diagram of the buccal and pharyngeal esophageal reflexes. The esophagus. Its superior muscularis is skeletal muscle for the voluntary control of the buccal esophageal reflex. Once triggered, the esophagus performs peristalsis to push the bolus to the stomach. Peristalsis is controlled by short reflexes at that point. The stomach Now the story of digestion becomes more complicated. Its activity begins in the cephalic phase and really speeds up in the gastric phase. It will switch its function in the intestinal phase. So, big picture. The stomach is responsible for chemical digestion and mechanical digestion. It receives a bolus, secretes and mixes in a special way by churning. Once finished, the bolus is liquefied into chyme, which begins to enter the duodenum. The stomach will secrete hormones for coordination of itself and some other organs. It also triggers a reflex called the gastrocolic reflex. When we look at the histology of the gastric mucosa, we see more complexity. There are several cell types in that simple columnar epithelium. These secretory cells are found in the deeper gastric gland, and their gastric juice rises through the gastric pit into the stomach lumen. You can see the cells listed here: parietal, chief, enteroendocrine, and even mucous neck cells. A list of cells and their functions. Mucous neck cells produce an acidic mucus to carry secretions out of the pits. Parietal secrete hydrochloric acid and intrinsic factor. The acid will denature proteins and assist with their enzymatic breakdown. The intrinsic factor goes into the small intestine and assists with vitamin B12 absorption. Chief secrete an enzyme precursor pepsinogen. It will be converted into the active form, pepsin, when it comes in contact with the hydrochloric acid. Enteroendocrine, also found in the duodenum. In the stomach, these cells secrete the hormones gastrin and histamine. With all that acid, the stomach can get down to pH 2 during digestion. So it needs mechanisms to protect itself from the acid. The goblet cells on the epithelial surface secrete a bicarbonate-rich mucus to neutralize a little of that acid. The columnar cells have tight junctions that are somewhat impermeable to the acid, and it's extremely regenerative. Functions of the stomach's local hormones. Histamine stimulates parietal cells even more. Gastrin stimulates parietal cells and stomach motility even more. This isn't positive feedback, but you can see how the stomach is receiving multiple stimuli, nerves and hormones. The stomach's function for digestion: liquefy the chyme. It does that by starting chemical breakdown of proteins with pepsin, starts breakdown of lipids with a gastric lipase, and mixes everything together by churning until it's thoroughly mixed and liquefied. To do this requires the initial closure of both sphincters. Once the chyme is formed, the pyloric sphincter opens and the stomach switches to peristalsis through the pyloric region. Pause here. Print this one slide on a single page and use it to review what we've learned so far. Think about food. Stimulates cephalic phase, which causes salivation and starts stomach secretion and motility. Swallow. Bolus in the stomach stimulates the gastric phase, which increases stomach activity. Liquefy the bolus and some chyme enters the duodenum. Stimulate intestinal phase, which increases intestinal activity and switches stomach activity. Now we can focus on the small intestine's contribution to digestion. The three regions are specialized: duodenum, jejunum, and ileum. Most absorption will take place in the ileum, but most of the chemical digestion will take place in the duodenum. Short and long reflexes control segmentation, then switching to peristalsis. And the intestine also releases hormone as an endocrine function for digestion. Motility. Segmentation is for mixing chyme with pancreatic juices. Peristalsis is for moving to the lower regions for chemical digestion and absorption, all controlled with local reflexes. The sequence of events in the small intestine. First, chyme gets mixed with pancreatic juice and liver products, enzymes and bile, respectively. These enzymes and the bile enter through the hepatopancreatic ampulla. The duodenum mixes everything with segmentation, then switches to peristalsis to move the mixed chyme forward. As it travels through the duodenum and jejunum, the enzymes break large molecules, or polymers, into small molecule subunits, or monomers. By the time the monomers reach the ileum, they're small enough to be absorbed by the simple columnar cells and placed into the capillaries within the lamina propria. We are about to look at the polymers, monomers, and absorption methods for these macronutrients. The polymer amylose, or starch. It is broken with salivary and pancreatic amylase enzyme into the monomer glucose. That is transported by facilitated diffusion to the capillary bed of the villi, then moved to the blood and transported throughout the body. The polymer protein. It is broken with gastric and pancreatic enzymes like pepsin, trypsin, and chymotrypsin into the monomer amino acids. These are transported by facilitated diffusion into the same capillary beds of the villi. A diagram showing you how the simple columnar cells move the monomers. The polymer nucleic acids, or DNA and RNA, similar to the others, broken with pancreatic, ribonucleases, and deoxyribonucleases into the relatively small monomer nucleotide. These are a bit bigger than glucose and amino acids, so they are actively transported into the same capillary beds. The polymer fats. These are the exceptions to the rule because they are big and non-polar. First, they're broken with lipase enzyme into a 3-carbon molecule called glycerol and the attached fatty acids using emulsification by bile. There's a picture of these on slide 36 and 37. Glycerol and small fatty acids are taken into the same capillary beds and processed by the liver, but longer fatty acid chains cannot cross into the blood. They aren't soluble enough. The columnar cells wrap them in a temporary transport protein to form a structure we'll call a chylomicron. That is moved into the lymphatic capillary structure, a lacteal, and into the liver where it is reprocessed. Now, if you're curious, it is the liver that creates LDL and HDL cholesterol. These are the fatty acids wrapped in more permanent transport proteins. The green portion is the short glycerol. The 3 long carbon chains are the fatty acids. Most fatty acids are anywhere from 14 to 24 carbons long. The fatty acid you eat determines whether the liver transports it as an HDL, good cholesterol, or an LDL, or bad cholesterol. Emulsification, another process needed for fats. Larger fat globules are separated from each other using bile salts. If we don't do this separation, we cannot digest fats efficiently. Imagine a small enzyme trying to chip away at that large globule. That would take forever. So bile salts are used to divide the large droplet into lots of tiny fat droplets. This allows for more surface area for the lipase enzymes, and fat chemical breakdown is much more efficient. Side note, fat is not bad for us. It's the type of fat we eat in connection with processed carbohydrates that is bad for us. An illustration of how the simple columnar cells create the chylomicron and use the lymphatic lacteals to transport it. We can now switch focus to the colon or large intestine. This organ is responsible for water and electrolyte absorption. It then prepares all the leftover waste, stores it, and removes it from the body. It is initially stimulated during the gastric phase by the gastrocolic reflex and eliminates the waste via the defecation reflex. Interestingly, it has two sphincter muscles. Internal anal sphincters are made of smooth muscle and under autonomic control, versus the external anal sphincter made of skeletal muscle and under somatic control. The colon contains a lot of bacteria, your normal flora. These bacteria have significant function. They ferment indigestible carbohydrates for us and as a result produce B complex and K vitamins for our use. They may also potentiate or train the immune system. Now, the byproduct of that carbohydrate fermentation can be gases. Flatus is the fancy name for fart. A list of the items absorbed by the colon. Most electrolytes are actively transported, and water is absorbed by osmosis. A diagram of the defecation reflex. Fecal material is slowly packed into the rectum. Once the rectum is full, its walls are stretched, and this triggers the reflex. The smooth muscle internal anal sphincters are opened, giving you the urge to go to the bathroom. Note that this is parasympathetic control. The skeletal muscle external anal sphincters allow you to wait until it's convenient, as they are under voluntary or somatic. And lastly, movement throughout the colon is by haustral contractions, kind of like a segmented peristalsis. Now for accessory organs. The liver is far more functional than we realize, including producing the bile that will be stored and concentrated in the gallbladder. Structurally, the liver is divided into lobules with triads of vessels. The lobule. Note the structure here. Several plates of cells surround a central vein. Blood would enter into the red hepatic artery and the blue hepatic portal vein structures, flow past the cells through the sinusoid capillaries, and out the central vein into the hepatic vein. You can see that all hepatocytes are in close proximity to the blood supply and those permeable capillaries. These same cells produce bile. However, that never enters the blood It follows the opposite route, entering the green bile duct and leaves the liver. A close-up view. Remember, the triad consists of the vessels, and bile flow is opposite the blood vessels. Liver sinusoids, highly permeable capillaries. They allow for the hepatocytes to easily secrete plasma proteins into the blood, as well as to process blood-borne nutrients. Kupffer cells are fixed macrophage for immunity. The hepatocytes have a lot of functions. Only a few are listed here. They produce the bile used in fat digestion. They process blood-borne nutrients. Remember how fatty acids are taken to the liver through lymphatics? The hepatocytes will unwrap the chylomicrons and repackage the fatty acid with different transport proteins. The liver can also change its metabolism according to what you eat. For example, you can eat no carbohydrates and survive just fine because your liver will make glucose for the rest of your body. They store fat-soluble vitamins A, D, E, and K, and they can break down some toxins from the diet. So what the heck is bile? I'm glad you asked. It's a cocktail of different things. So really, bile is part of bile salts. They take large fat droplets and break into smaller fat droplets. Again, not chemical digestion, but more of a mechanical digestion. We're not breaking bonds. We're making the glob of fat smaller so that the enzymes have more surface area and can act faster. Bile salts are recycled in the enterohepatic circulation. So the hormone CCK is responsible for fat digestion. When fatty chyme enters the duodenum, the enteroendocrine cells there release CCK to the bloodstream. CCK opens the hepatopancreatic sphincter, causes contractions to occur in the gallbladder, and increases liver production of bile. CCK also stimulates lipase-rich pancreatic juice production by the pancreas. Okay, a mini case study for you. Use what you've learned about the liver, bile production, and even plasma proteins to figure out this case study. In the A&P Cafe, explain these symptoms. A person suffering these symptoms in the previous slide. A picture of the liver whose hepatocytes are dying and being replaced with fatty scar tissue on the top right. A picture of a liver scarred from cirrhosis. Now, I've been told by nursing faculty that you will most likely, on a clinical, be shown a patient with these symptoms. Be able to explain why the loss of liver function is causing these symptoms. The pancreas, an interesting organ because it is both endocrine, secretions released to blood, and exocrine, secretions to the body surface. A diagram of pancreatic acinus. The cells within this structure, acinar cells, secrete bicarbonate and enzymes into the small duct. These will eventually end up in the main pancreatic duct, where they will enter the duodenum by the hepatopancreatic ampulla. Most pancreatic enzymes are released to the duodenum in an inactive form. Again, most pancreatic enzymes are released to the duodenum in an inactive form. They will become activated in the duodenum. Those listed here, trypsinogen and chymotrypsinogen, digest proteins once activated. The pancreas also secretes amylase and lipase, and bicarbonate neutralizes the acid in chyme. A list of pancreatic enzymes and how they are regulated. Remember, CCK predominates when you eat a fatty meal. A normal pancreas at the top versus a cancerous pancreas at the bottom. Now for a topic switch. You have the basic functionality of alimentary canal and accessory organs, including the main reflexes and enzymes. You also now know that digestion is divided into 3 phases: cephalic, gastric, and intestinal. Additionally, you know that each organ is primed before the food arrives at it. But now we need to discuss the digestive system's relationship to blood pH. There are pathologies where too much bicarbonate is lost from the body. Remember, that serves as a buffer. When we lose bicarbonate, as in diarrhea or renal loss, we lose our buffer and become acidic. Metabolic acidosis also occurs during diabetic ketoacidosis, renal failure, and intoxication by methanol and ethylene glycol. These are all deadly situations. Diagram of causes and warning signs of metabolic acidosis. When we lose too much hydrogen ions from the body or have too much bicarbonate, we experience metabolic alkalosis. Vomiting causes a loss of our stomach acid. Certain diuretics cause us to secrete hydrogen ions. On the flip side, taking too many antacids in a day is really a bicarbonate overdose. Diagram of causes and warning signs of metabolic alkalosis.